In this podcast, we'll do a tour of the cell, looking at its structure and how it works. But first, here's a few things you need to know about cells. First, all organisms are composed of cells, one or more. A bacterium is made of one single cell. A human being is made of something like 40 trillion cells. That's 40 with 12 zeros. Cells are also the basic functional unit of all organisms. Processes associated with life are performed by cells. For example, cell respiration, muscular movement, or even the act of thinking. Finally, cells come from pre-existing cells. All your cells come from several cycles of division, starting with one single cell called the zygote. The zygote itself came from your mom's egg cell and your dad's sperm cell. In this podcast, we'll look at the structure and function of the main components of a eukaryotic cell. We'll take the animal cell as an example, but we'll also look at some specific components of plant cells. Let's begin with the outer layer of the plasma membrane. The membrane is composed primarily of phospholipids arranged in a bilayer. The hydrophilic heads of the phospholipids are on both sides of the bilayer, and the hydrophobic tails are in the middle. Proteins are either attached to the surface of the membrane, these are called peripheral membrane proteins, or embedded within the membrane. These are called the integral membrane proteins because they're an integral part of the membrane. In animal cells, cholesterol is also found in the plasma membrane. Its main function is to stabilize the membrane when temperature changes. The outer or extracellular side of the membrane also has glycoproteins and glycolipids. That's carbohydrates attached to proteins or lipids. These molecules represent a sort of chemical signature for the cell and are involved in cell recognition and signaling. If we move into the cell, we see several organelles found in the cytoplasm or living space of the cell. We'll now look at the structure and functions of these intracellular components. Keep in mind that these organelles work together. They're functionally related, as we'll see next. Let's begin with the nucleus. That's the headquarters of the cell. The nucleus contains your DNA arranged in the form of chromosomes. The nucleus is a fortress surrounded by two membranes. That's two phospholipid bilayers. We call this the nuclear envelope. There are pores in the envelope to allow the traffic of molecules such as messenger RNA, which is a transcript or a copy of a gene that will be used to make a specific protein. In the nucleus is an area called the nucleolus. This nucleolus is used to synthesize ribosomes, which are the protein factories, as we'll see a bit later. Ribosomal subunits are made in the nucleolus and are then shipped to the cytoplasm through the nuclear pores. In the cytoplasm, ribosomes will assemble and will be ready to translate messenger RNA into proteins. Ribosomes are of two types. There's the free ribosomes. They're found in the cytoplasm and they make proteins that will stay in the cytoplasm. These ribosomes are shown as small dots here. Bound ribosomes are attached to the outer membrane of the nuclear envelope or to a connected structure called the endoplasmic reticulum. These bound ribosomes make proteins that will be exported from the cell or sent to membrane-bound organelles. Let's look at this closer view where we see the nucleus, the ribosomes, and the endoplasmic reticulum in more detail. Now, the endoplasmic reticulum is a network of bags and tubes made of membranes, and it's continuous with the outer membrane of the nuclear envelope, as we can see here. There are actually two types of endoplasmic reticulum. The smooth smooth endoplasmic reticulum, is smooth because it doesn't have ribosomes attached to it. The smooth ER is involved in the synthesis of various lipids, the storage of glycogen as an energy reserve, and the detoxification of some drugs. The rough ER is said to be rough because it's covered by ribosomes. Of course, this rough ER will be involved in protein synthesis since it's covered by ribosomes. Remember that the so-called bound ribosomes are attached to this rough ER. These bound ribosomes make proteins that will be exported from the cell or sent to membrane-bound organelles in the cell. Here's how this works. The protein made by the bound ribosomes are incorporated in the lumen or internal space of the rough endoplasmic reticulum. Then the protein is packaged into a vesicle, which is a bag of membrane that buds off of the rough ER. This vesicle moves towards another organelle called the Golgi apparatus, which is a sort of shipping and receiving department for the cell. The Golgi will modify and direct proteins to their final destination. So the vesicle carrying the protein fuses with the cis or receiving face of the Golgi apparatus. In the Golgi, the protein will be modified as it moves towards the trans or shipping face of the Golgi. Finally, the modified protein will leave the Golgi in a vesicle and reach its final destination. For example, a protein that will be secreted or exported from the cell will travel in a vesicle that will fuse with the plasma membrane. This will release the protein in the extracellular space. A protein can also be sent to membrane-bound organelles in the cell. For example, lysosomes are membrane-bound organelles that receive their proteins from the Golgi. These proteins found in the lysosome are hydrolytic enzymes, which means that they can hydrolyze organic molecules. With these hydrolytic enzymes, lysosomes are involved in intracellular digestion. They can digest food particles in some unicellular organisms. Lysosomes can also digest and break down foreign particles in cells that were ingested by your white blood cells, or they can be involved in the recycling of your own damaged organelles. The nuclear envelope, the endoplasmic reticulum, the Golgi apparatus, the lysosomes, and the vesicles are all part of what we call the endomembrane system, which means membranes inside the cell that work together. Another component of the endomembrane system is the vacuoles, which are membrane-bound bags larger than vesicles. Vacuoles actually play a variety of functions in different organisms. For example, in plants, a large vacuole called the central vacuole often fills most of the living space in the cell. This central vacuole can hold food reserves, waste, pigments, or poison. Another organelle found in both plants and animal cells is the peroxisome. Peroxisomes are used to degrade certain molecules such as poisons by oxidizing them. This oxidation produces hydrogen peroxide, which will be neutralized in the peroxisome. Two of the most important organelles are the energy transducers, the mitochondria and the chloroplasts. Their main function is to transform energy from their surroundings into a usable form of energy. In plants and all other photosynthetic eukaryotes, the chloroplast is the site of photosynthesis. The chloroplast is surrounded by two membranes. In the chloroplast are flattened bags of membrane called the thylakoids. They're often stacked together to form a granum. These thylakoids contain the photosynthetic pigment chlorophyll and are responsible for converting light energy to chemical energy, which is the first step of photosynthesis. The rest of the reaction involved in photosynthesis occur in the stroma, which is a fluid found in the chloroplast. In both plants and animals, the mitochondrion is the site of cellular respiration. The mitochondrion is also made of two membranes. The reactions involved in cellular respiration happen in and across the inner membrane. To maximize the rate of the reactions, this inner membrane of the mitochondrion is convoluted and forms several folds called cristae. The entire cellular structure is supported by a network of protein fibers called the cytoskeleton or cellular skeleton. The cytoskeleton is also involved in movement. For example, vesicles that travel from the rough endoplasmic reticulum to the Golgi apparatus are transported along cytoskeletal fibers called microtubules. Cytoskeletal fibers are also responsible for the movement of cilia and flagella, such as those that propel sperm cells. The cytoskeleton is also responsible for muscle movement, and it's involved in the movement of chromosomes during the processes of mitosis and meiosis. The cytoskeleton is anchored to membrane proteins, which are themselves attached to the extracellular matrix, a network of proteins such as collagen and elastin that support cells and allow adhesion of cells. I hope you found this podcast useful. Don't forget to try the quiz on the blog. The address is biopodcasts.blogspot.com. Talk to you soon.